All right. So the second class that we will be talking about اللي هم الماكرولايدز. من اسمهم؟ How can you relate the name to the structure؟ ما نحكي macro. Yeah, macro is a, let's say, prefix which states that it's a huge, let's say, structure. عفوا. طيب تمام. And macrolide, because let's say it's a huge lactone structure. As you can see, it's a cyclic compound that is composed, or that is, let's say, has a cyclic ester. That is why it's a lactone. It's a macrolactone or macrolide. All right. Do you think it's a natural compound or something that was synthesized in lab? Based on the complexity, so many stereocenters that we see in the structure of this representative macrolide, it's definitely a natural compound. Now, this macrolide, or macrolides in general, these are composed of different numbers of the, or number of atoms that constitute them ranges from 14 to 16. Okay? يعني لو بدي أعد the number of atoms ممكن يكون 14 ممكن يكون 16, and we will say state the exact number for each of the macrolides that we will be talking about. The members, by the way, in this class are very يعني ما بيجوزوش إلا عدد بسيط. يعني they do not, let's say, أو ما بيجوز عددهم 5. لكن in terms of, let's say, the structure, what combines them, and they have a huge, a large cyclic structure that has amino sugars bound to it. The amino sugar is called disosamine, and as you can see, it's a cyclic or a cyclohexane that one of its atoms is an oxygen. One of its hydroxyls is replaced with a dimethyl nitrogen, and the other sugar is called cladinose. Right? Right. Do you think this compound is an ionizable compound? What is the ionizable functionality in its structure? Yes, this dimethyl nitrogen, or this tertiary amine, is an ionizable or is an aliphatic tertiary amine that has a pKa around 8, which means it is an ionizable functionality that can be ionized at physiological pH with a certain percentage. برضو كمان, this allows us to formulate salts out of it. Or in other words, I can generate salts out of this compound, يا إما to increase or decrease in water solubility depending. Okay, لكن overall, do you think this compound has high water solubility? What do you think? في hydroxyls, لكن في المقابل, yeah, we have a lot of, let's say, carbons. We have a lot of methyls, ethyls, on the macrocyclic structure, and even on the sugars themselves. But all in all, this compound is considered, let's say, not highly water soluble. يعني صح في عندي hydroxyls, لكن في المقابل I have so many carbons, aliphatic carbons, that are considered lipophilic in nature. حتى بالionization definitely أنا بزيد عندي الwater solubility. أو إذا أنا بحوله لسولت بخليه بهاي الحالة water soluble depending كمان على الكاونتر ion. We will see examples on that. لا مش شرط, يعني حتى لو تطلعي على هالsugar for instance, this one صح في عندي hydroxy بس في عندي methyls على سبيل المثال. Okay, we have a methoxy in here. Here we have a methyl. It's lacking the hydroxyls. يعني صح في عندي an ionizable group, لكن if you consider the rest of the structure, it has so many methyls and an ethyl group, so many carbons. All in all, the compound has way more lipophilic groups than this ionizable group, أو حتى كمان الهيدروكسيلز all together. Okay. طيب, as I've mentioned, بما إنه we have an ionizable group, معناها إنه we can generate salts out of this, let's say, compound. هلا depending على الكاونتر ion, its solubility in water يا إما بتزيد أو بتقل حسب. Okay. هلا if the counter ion is glucoheptanoic acid, do you think such a salt will increase or decrease in water solubility? أكيد increase. Okay. This carboxylic acid, when deprotonated, having an ionic interaction with the protonated nitrogen, look at the rest of the carbons, all of them have hydroxyl groups. For all in all, this will increase in water solubility for the macrolide. طيب اللاكتوبيونيك acid برضو كمان such a counter ion or a salt that has of the macrolide that has the lactobionic acid for sure will also have higher water solubility. طيب what about these salts? These counter ions, if you would, let's say, generate lauryl sulfate salt of the macrolide, would it be more water soluble or less? Less. It will be highly lipophilic in that matter. I will increase in water lipophilicity way more. طيب this compound, or if you look at the, let's say, pharmacophore of the macrolides, okay, can you generate, or can you, for instance, by looking at this structure, إنه ممكن إحنا نحدد إيش في عندي repeating units in this structure? يعني لو for instance طلبت منكم إنكم تفكروا what would be the building block in this matter for the macrolides? Forget about the functionality. The skeleton of the building block will be composed of how many carbons, you think? 3. Yeah, actually, macrolides are biosynthesized from propionic acid, which is composed of, let's say, 3 carbons and a carboxylic acid. Okay, if you look at the structure, you can, or as you can see in here, we have alternating 3 carbon skeletons that are linked together via covalent bond. But this is, أو خلينا نحكي, any of those is considered one of the building blocks, okay? And that's the precursor from which the natural source produces the macrolides. Later on, it eventually oxidizes 2 of those carbons, and then eventually we get the structure of the macrolides. And later, 2 sugar units are added to them. Okay? Okay. One of the issues that is associated with erythromycin, the first macrolide that was isolated, إنه كان acid unstable. هلا they found out, the reason for that اللي هي الهيدروكسيلز اللي موجودة عندي on let's say those carbons in here. تمام؟ This functionality is called what? Ketone. شو بتعرفوا عن الكيتون in terms of electrophilicity؟ يعني لو بدي أقارنه مثلا بأمد for instance, أو even الesters. More electrophilic, okay? Because I have carbons, أو يعني if I would compare it to amides and esters, here we have a heteroatom. صح بتسحب electrons inductively, لكن at the same time it's donating electrons via resonance. Here we do not have anything that is donating electrons via resonance to this carbonyl carbon. That is why this carbonyl carbon is considered an electrophilic carbon, electrophilicity even higher than amides and esters. Now, in the structure of macrolides, those oxygens on carbon 6 and 9 of those hydroxyls, the oxygens يعتبروا nucleophilic. فاللي بيصير عندي in acidic conditions, this carbonyl oxygen can get protonated. And if protonated, this will increase or decrease electrophilicity. It will increase it, لأنه أنا صارت عندي هون after protonation, الأكسجين will be positively charged, and being positively charged معناها رح يصير withdrawal via the inductive effect أكثر، ورح يزيد عندي electrophilicity أكثر وأكثر, which will make it more prone to attack by nucleophiles. Okay, since إنه in the structure طبعا هاي أحيانا ما بيوحي زي ما لما نرسم هيك in a 2D representation إن ال OH بتكون عندي قريبة من الكربونيل كربون، لكن أنا لو بدي أطلع على 3D geometry of this molecule, it can get to a point where the hydroxyl رح تكون عندي بالtrajectory الصح إنها تعمل attack على الكربونيل كربون. Okay, so what will happen simply إنه that oxygen, one of its lone pairs, will attack الكربونيل كربون. Okay, خلينا نرقمهم عشان لما نطلع على الانترميديت نعرف exactly what bonds were broken and what bonds, let's say, that were formed. نرقم هاي 1, 2, 3, 4. Okay, so this is 1, 2, 3, and 4. Okay, that's the carbon that had the double bond. Now when the oxygen attacks it راح تنكسر عندي الدبل bond وهاي الكربون راح تصير عندي quaternary أو sp3 hybridized. This is the here on carbon 1 we already have this bond with the hydroxyl and it has formed this new covalent bond بينها وبين الcarbonyl carbon which is numbered here 4. And that's, let's say, the oxygen that is already attached to carbon number 4. Okay? كلنا بنعرف لما يكون في عندي an sp3 hybridized carbon that has heteroatoms, all right, it's, let's say, unstable. Such a compound will tend, let's say, to يعني يا إما it will break one of its oxygen carbon bonds أو بحالة المacrolides this carbon being electrophilic لأنها right next to two oxygens withdrawing electrons inductively from it, this oxygen in here that is close to it will now, طبعا before the attack, sorry أنا كان لازم هاي وضحتها before, because of the acidic conditions ممكن يصير في عندي elimination. Any basic molecules موجودة عندي في المixture such as even the water molecules themselves can abstract that proton وبتنكسر عندي هاي الباند وبتطلع ال OH وبتتكون a double bond between carbon 3 and 4 which will make even carbon 4 more electrophilic subject to attack from the nucleophile on this carbon. Okay, yes. عفوا. هلا الباند اللي بتنكسر اللي هي الباي باند ما بين carbon 4 والاكسجين. اوكي؟ مقابل الباند اللي راح تتكون ما بين الاكسجين that is on carbon 1 and carbon number 4. يعني لو نرجع لهالستركتشر that's the new bond that is formed and الدبل bond اللي كانت ما بين هاي الكربون وهاي الاكسجين هي اللي انكسرت. Okay? Any questions so far? Yes. هلا generally speaking لما يكون في عندي withdrawal or donation by resonance بيكون احيانا او in most cases it would be more profound. The resonance effect usually بيكون عندي more profound unless إذا كان ال environment around that atom that has for instance heteroatoms اللي بتسحب لها electrons يعني ال donation via the sigma bond فقط هي الكربون اللي بتعمل donation via the sigma bond. اوكي فاحنا يعني لما بدنا نقارن او لما بدنا نشوف انه خلينا نحكي الاندكتيف effect you usually think about atoms that are withdrawing electrons. اوكي لكن in general احنا بنعتبر انه ال resonance has a more profound effect relative to the inductive effect unless I have what is attached to the carbon that I'm concerned about many heteroatoms that are withdrawing electrons. بهاي الحالة ممكن انك تاخذ بعين الاعتبار انه الاندكتيف effect بيكون عندي more profound. Okay? فيعني it's case by case. I would يعني when thinking about it بدك تطلع على so many factors. هلا مثلا على سبيل المثال انت عندك ما بين الاميد وما بين for instance الاستر. مين easier to hydrolyze؟ الاستر. Why؟ هلا شو الاختلاف ما بين النيتروجين والاكسجين؟ الالكترونيجاتيفيتي رايت مين مور الكترونيجاتيف؟ الاكسجين. هي صح بتسحب الكترونز alright يعني في عندك two opposing effects بهاي التو molecules الاكسجين بتسحب الكترونز inductively alright لكن at the same time عم تعطي electrons via resonance نفس الشيء هون. هلا بما انه هاي more electronegative اسفه بما انها less electronegative النيتروجين معناها سحبها للالكترونز inductively بيكون اقل والدونيشن فايا الكترونز بيكون افضل ولا اقل من الاكسجين؟ بيكون اعلى لانها less electronegative عندها قابليه انها تعطي الالكترونز بشكل اسهل من الاكسجين. But when you let's say consider those two factors together بهاي الحالة أنا طبعا definitely يعني for E يعني for خلينا نحكي يعني لكل واحد لحال. لو أنا بدي أخذه بعين الاعتبار إنه أنا في عندي آتوم عم تعطي إلكترونز via the induction أو آسفة via resonance، معناها هي عم بتقلل من الإلكتروفيليسيتي، لكن at the same time هاي الآتوم عم تسحب إلكترونز من الكربونيل كاربون، فهي عم ترفع الإلكتروفيليسيتي. لكن ما بين those two factors مين اللي more profound؟ الريزونانس. بيكون المقارنة أصح لو أنا مثلا بدي أقارنها بالدهايد أو كيتون على سبيل المثال. أوكي؟ هلا ما في عندي الكربون صح تعتبر إلكترون donating inductively، لكن ما في شيء عم بيسحب، لا هي عم تعطي الإلكترونز باي ريزونانس ولا عم تسحب إلكترونز باي ريزونانس. عشان هيك بنعتبر إحنا البارشل بوزيتيف تشارج اللي عليها بتكون much higher than the amide than the ester. أوكي؟ طيب هلا this is the reason for those two hydroxyls along with this carbonyl, the reactive carbonyl, those are the reasons for the acid instability of macrolides. وin particular طبعا مش كل الماكرولايدز، لانه later generations صار فيهم modifications تمنع هالشيء. اوكي لكن خلينا نحكي for the first one that was isolated اللي هو الاريثروميسين، the acid instability is because of the interaction اللي بيصير او intramolecular ketal formation او اللي انه الهيدروكسيلز are attacking الكربونيل كاربون. اوكي طب كيف انا ممكن احل هيك مشكله بالاريثروميسين؟ How? Yeah, this انه let's say to change the route او for instance انه tablets او the drug to be given او يعني as in the form of an enteric coated tablet بقلل exposure to acidic conditions. This is one way. طب structurally لو انا بدي احسن او خلينا نحكي امنع هالشيء انه يصير. لا yeah yes انه one of those hydroxyls to remove them او as you'll see later on to mask them to lower the ability for them to attack. هلا yeah if you would prevent أو if you would let's say remove one of those hydroxyls حتى لو تكون عندك let's say this compound وارد جدا يعني مش مبين عندك هون في السكيم وارد جدا إنه يرجع هاي الأكسجين let's say اللون pair of electrons ترجع وتكون double bond وترجع تكسر الكمباوند ويرجع let's say to its form. Okay, لكن this is a reversible step. لكن if it will let's say move on all the way to the ketal formation, this is not a highly reversible step. وadd to that إنه ممكن تكون subject للattack by water. Okay. That's enough for خلينا نحكي stabilizing the compound أو the drug in acidic conditions. Okay. طيب. نحكي هلا عن الميكانيزم اوف اكشن. يعني صح بالامتحان مش مطلوب منكم تعرفوا كل الديتيلز اللي انا راح احكيها هون، بس يعني it's good let's say to understand it. طيب what is this crystal structure you see in front of you? That's the ribosome. That's the 30S unit and this is the 50S unit. Okay? الماكرولايدز بيرتبطوا بال 50S. طيب what do you think these structures are اللي موجودين عندي في البلو والاورنج؟ Can you be more specific? أي RNA ولا transfer RNA? Okay, these are the RNA molecules that brings in the amino acids which are represented here by let's say those spheres. فاللي بيصير عنده إنه messenger RNA that binds to 30S is being read by the ribosome where the codons in the messenger RNA are translated into amino acids. الماتش ما بين الكودون والانتي كودون which is proofread in the 30S subunit is what eventually decides what amino acids to add to the growing chain of the polypeptide. لاحظوا انه بصير في عندي خلينا نحكي synthesis of that polypeptide chain in the 50S unit where it has a passage from which it leaves the 50S subunit. Okay? طيب هلا to link the coming amino acid to the already built polypeptide chain يعني هاي إشي زيادة بتحكيه بس أكيد يعني don't worry about the detail at all. The amino acid is linked via an ester bond to the nucleotides that are part of the transfer RNA. Okay, here's the polypeptide that is being synthesized, which is called, before being folded, a nascent peptide. Okay, هلا it's already bound to the transfer RNA اللي كان before via an ester bond. فاللي بيصير إنه the coming amino acid on transfer RNA اللي بيكون عندي موجود بيسموها في pocket A، النيتروجين اللي هون attacks the ester, which will form a peptide bond between this newly coming amino acid with already growing polypeptide chain. Okay, هلا الماكرولايدز بيرتبطوا at the entrance or very close to where this process occurs. يعني if we the green spheres that you see in here, or this green molecule you see in here, represents the erythromycin. لو بدي أعمل zoom in, this is where خلينا نحكي ال A will be sites for the two transfer RNAs that are being bound to the ribosome. This is where the erythromycin will be bound, very close to them. All right. طيب هلا لو بدي اخذ، او لو افترضنا انه انا بدي اطلع from, let's say, this angle, as if I am looking at the tunnel from which that polypeptide is coming out. Look at where erythromycin is. It's in this tunnel, preventing, or it eventually disrupts خلينا نحكي the growth of that polypeptide chain. يعني بيكون موجود عندي في التانل وبيعيق عملية اللي هي انه يطلع عندي البوليبيبتايد chain وحتى يصير في increase بالlength of that polypeptide. Okay, and this is how it prevents protein synthesis. It binds to the 50S subunit, very close to where those amino acids are being linked to form the nascent polypeptide. At the entrance, أو خلينا نحكي at the tunnel from which this nascent peptide leaves, the erythromycin binds there, and it prevents the leaving، أو خلينا نحكي إنه يصير في growth لهاي البوليبيبتايد chain. يعني eventually it will disrupt any protein synthesis after it binds to the 50S unit. Okay. وضحت؟ لا هو ما هو بيرتبط قريب منهم، بس ما بيرتبط محل الترانسفير RNA، بس بيرتبط يعني لو يعني again if this is the tunnel from which that polypeptide chain, that's the peptide that is being synthesized، الإريثرومايسين بيرتبط بهاي التانل very close to it، فبيعيق إنه هاي البوليبيبتايد اللي عم تتكون إنها تطلع من هاي التانل. و if it is stopped, okay, this will disrupt the binding of the transfer RNAs، إنه يصير في عندي any conjoining ما بين الأمينو أسيدز ما بين الترانسفير RNA اللي مرتبط في ال A site وفي ال P site. Okay, this is way more detailed than you need to know for this course. بس at least يعني بتعطيكم فكرة لما بدكم تتذكروا mechanism of action of macrolides، إنهم وين بيرتبطوا eventually what exactly it does. Okay. Yeah, this is the peptide. This is the nascent peptide that is being synthesized. Okay, erythromycin binds at a site very close to them. So بيكون كمان very close to where the transfer RNA is. As you can see, وكأنه بيعمل bulkiness in that tunnel, which will prevent إنه nascent peptide إنه عم بيطلع من عندي هاي التانل. وبكذا of that ما بصير في عند وكأنه بيوقف إنه يصير في any binding ما بين الامينو acids that are being brought to by the transfer RNA to the already growing chain. اوكي، وكأنه بيوقف عملية انه البيبتايد عم بيطلع، وبالتالي بيوقف عملية انه يصير في عندي any growth of that polypeptide chain that is being synthesized. Okay, they did what is known as Western blots. يعني هاي بس هيك صورة زيادة افرجيكم اياها. For the protein synthesis before and after in a bacterial cell, before and after the binding of a macrolide. كل let's say هاي الblue dark dots that you see represents the different proteins that are being synthesized in a cell. After the binding of macrolides to the ribosome، لاحظوا كيف الإيمج تماما عندي اختلفت. يعني it inhibited the synthesis of a lot of proteins that are essential and functional to the cell. اوكي. هلا مكتوب عندكم في السلايدز انه الماكرولايدز are bacteriostatic and they are actually او first generation are. لكن في later generations رح نحكي عنهم او رح we will see an example for them. They are actually considered bactericidal. لانهم لما يرتبطوا بالرايبوسوم بيكون ارتباطهم جدا 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 قوي. Dissociation جدا ضعيف. So they inhibit the synthesis of so many proteins that some of them is very essential for the survival of cells. فيعني إن نحكي الماكرولايدز in general bacteriostatic, this is not an accurate word. It depends على الماكرولايد والresidence time of it inside the ribosome. إذا بيرتبط بشكل جدا قوي والديسوسيشن بيكون ضعيف هاي بيعتبر bactericidal. إذا ارتباطه يعني قوي بس مش as strong as for instance التليثرومايسين والسيليثرومايسين, it's actually a bacteriostatic. فit depends على concentration, بيعتمد على الدرug eventually إنه how strong it binds or how strongly it binds to the ribosome. Okay? طيب هلا بما انه احنا عم نحكي عن او بكل انتي بكتيريال كلاس عم نحكي عن الريزيستنس associated with them, فرح نحكي بس عن the specific resistance that was developed against the macrolides. The most important one اللي رح نحكي عنها اللي هي الميثيليشن of adenine او conversion او mutation of adenine into the corresponding guanine. Where macrolides bond, in this image, this represents one of the macrolides, which is telithromycin. There is بيكون عندي في pocket where they bind in adenine, right? Some bacterial strains had mutations where they would change adenine into guanine. That specific mutation is called A2058G. يعني إنه تغير عندي ال A to G وهي sequence number of that adenine. هلا if you look at their structures بإيش يختلفوا عن بعض؟ الأدينين والجوانين. إنه هسه صار في عندي بهاي البوزيشن instead of hydrogen we have an amino group. مين bulkier? Yes, so the guanine is a bulkier nucleotide relative to adenine. And because of that, because of this extra amino group that was, let's say, introduced or when changing the adenine into the guanine, that amino group have led to clashes ما بين الماكرولايد لما يرتبط محل ما هو أصلاً بيرتبط وما بين هاي الجوانين. This will highly lower the binding affinity of the macrolides to those ribosomes اللي فيهم هالmutation. تمام؟ Single mutation غيروا الأدينين into guanine, صارت الجوانين bulky, قللت من size of the binding pocket of the macrolide, and that is why the binding affinity has been decreased massively, which led to resistance against the macrolides. What matters to me in this slide, in the macrolides may undergo metabolism where one of the major metabolite is where one of the methyls on this nitrogen undergoes a demethylation. هلا عن each number of the members of the macrolides, and we will start with erythromycin ورح نحكي specifically عن their esters and salts. Erythromycin, which has this structure, has a site for salt formation, and also this hydroxyl can be converted into the corresponding ester. ودepending على the group that is attached to the ester group يا إما بتزيد الwater solubility أو بتقل. فرح نحكي عنهم one by one. Erythromycin estolate. This is what? A salt derivative of erythromycin or an ester. It's actually both. Okay, the oxygen has been, let's say, attached to this acyl group via esterification, and also that nitrogen is bound via an ionic bond to the lauryl sulfate. هلا both of what is the counter ion of the salt will ester that was added will increase or decrease the lipophilicity. It will highly increase the lipophilicity. Okay, mostly this is for it will highly increase the lipophilicity. طيب, what about the ethyl succinate? Yep. Okay, that group or that ester that is added to the this amino sugar will increase the lipophilicity. Erythromycin stearate. This is the stearic acid. This is to generate a salt, you think, or an ester? A salt. Okay, it's actually generated as a salt. More lipophilic than erythromycin or less? More, for sure. Okay, erythromycin lactobionate. It's to generate a salt, and it will increase or decrease lipophilicity. Decrease. It's actually for the reason of increasing in water solubility. In the case of if I need, let's say, to administer erythromycin in a salt or in a highly water soluble form, as for parenteral administration. تفضل. Mostly for masking the taste. Macrolides has a very better taste. طبعا مش انه بيأثر على الابزوربشن بشكل عالي، يعني still it has good absorption. لكن it's mostly to mask the taste. It has a very bad taste. طيب, these are the rest of the members of the erythromycin. يعني هاي تقريبا آخر سلايد رح نحكي عنه. Look at clarithromycin, and if you have your notes for the erythromycin structure, compare the two structures together. Where's the variation between them? Yes, here, as you can see, the hydroxyl on this position has been converted into a methoxy. طب هاي كيف ممكن تأثر على الacid stability of clarithromycin? Yes, because as you know, this oxygen is involved in the formation eventually of the ketal. هلا لما يعني صح ممكن هاي الأكسجين تعمل attack عندي هون على الكربونيل carbon, all right? لكن يعني something probably I did not explain in the previous one. Let me just go back to it. احنا usually بالmechanisms we do ignore البروتون swap. بس هاي الستep أول ما بتصير بيكون أنا already عندي البروتون still attached in here and this one is positively charged. Okay, this is a stable intermediate relatively speaking لأنه ممكن any base موجود عندي في الميكستشر ياخذ هالبروتون ويعطيني this neutral intermediate. Okay, لكن in case كان عندي a CH3 that is bound to it, it may attack الكربونيل carbon. لكن this intermediate هل ممكن أنا آخذ هال-CH3 وأعطي هاي الإلكترونز to the oxygen? Is CH3 plus a good leaving group? إطلاقاً. Okay, فonce you methylate it مش إني أنا عم بقلل، يعني بقلل النيوكليوفيليسيتي لأنه صارت الأكسجين bulkier، لكن الأساس إنه when you convert the proton into a CH3 معناها if that oxygen is to attack الكربونيل carbon إذا عليها CH3 ما راح يكون في مجال إني أنا to let's say attack the CH3 وأكسر البوند ما بين الكربون والأكسجين. This is a strong bond that cannot be easily broken. Okay, يعني مش بسهولة ممكن أنا أعمل obstruction ل CH3 versus obstruction ل proton. There's a huge difference between them. فعشان هيك when you only methylate the oxygen of that clarithromycin, by that you will highly decrease the rate of ketal formation أو acid instability coming from the attack of this oxygen على الكربونيل carbon. All right. طيب الازيثروميسين. What is the variation ما بين الازيثروميسين والكلاريثروميسين والإريثروميسين? Yes. هلا the number of the atoms comprising this ring is more، يعني وكأني أنا شلت الكربونيل بدلت محلها نيتروجين وزدت كربون. Okay. Now يعني بالستركتشر إذا مرسوم عندكم بحيث إنه مرسوم بهاي الطريقة. This is wrong. There are oxygens in here. It's not carbons. Okay, just fix that. Okay. Yes. Yes, that's what they did. This is a semi-synthetic compound. Azithromycin is synthesized from clarithromycin. from erythromycin. طبعا مجرد اني انا شلت الكربونيل، حتى لو هاي الهيدروكسيدز I kept them the same، ما راح يصير في عندي any let's say ketal formation or inactivation via this mechanism. اخر compound راح نحكي عنه اللي هو التليثروميسين. What are the chemical changes in its structure? ايش هو عفوا؟ Yeah, they've conjoined the hydroxyls in here via a let's say a carbamate cycle and attached to this nitrogen, an imidazole linked to a pyridine. What else? The carbonyl is still the same. Is it acid stable? Because this oxygen is methylated, all right? Yeah, here they removed one of the sugars, the cladinose, and the hydroxyl in here is oxidized into the corresponding, let's say, ketone. Okay? Yes. Yes, the clarithromycin because of the methyl added to the oxygen. الازيثروميسين لانه it lacks الكربونيل. Same thing for التليثروميسين. The oxygen in here is methylated. التليثروميسين has a خلينا نحكي very strong binding to a ribosome. Mostly because this extra group in here binds into خلينا نحكي a cleft inside the ribosome which increases the binding affinity to this drug to the ribosome. So that is why it's a خلينا نحكي in terms of the activity it's more active than azithromycin than clarithromycin والerythromycin. Like the issue associated with this compound is that the pyridine in here, okay, has been associated with the liver toxicity seen with telithromycin. So that is why, let's say, in later generations of the macrolides, they've substituted the pyridine with a amino with an aniline ring, which is much less toxic, or does not, let's say, have the tendency to cause the liver toxicity associated with having the pyridine. Okay. Here, as you can see, it's just a crystal structure of erythromycin and telithromycin bound to the ribosome. They are superimposed on top of each other. بس لاحظوا أنا عندي هاي two ring systems that are found in telithromycin اللي موجود عندي in blue. As you can see, they can even bind deeper inside their binding site, forming more interactions, increasing the binding affinity. فاكشلي التليثروميسين يعتبر bactericidal macrolide versus الاريثروميسين اللي يعتبر bacteriostatic. Okay? Any question up to this point? Yes. هلا this is via experiments. يعني it's not, let's say, يعني it's not something that you can predict easily. Exactly ايش هي الاكسبيرمنتس done via which يعني the in vivo, let's say, studies that they've done where they linked البيريدين I'm not sure, بس my guess انه ممكن جربوا خلينا نحكي many يعني بالاخر التليثروميسين اللي طلع معاهم is a consequence of synthesizing many analogs. Probably when they compared it to other analogs that did not have that pyridine, ممكن كان محلها phenyl. This is how they linked liver toxicity to the presence of that pyridine. That's my guess. Yes. Ala? Hala, it could be that they've done that. يعني I'm not aware of any paper بالliterature أو حتى في patents where they made changes على the sugar part itself. بس يعني تنسوش طبعا بهاي باي ذا واي الجروبز اللي احنا عم نحكي عنها، الممبرز قليلين جدا مقارنه مثلا باللي شفناه في البنسلينز والسيفالوسبورينز، لانه as you can see this compound is very let's say complicated. It has so many functionalities. التوتال synthesis جدا صعب، وحتى لو انا عندي natural compound اني انا اعمل عليه اي modification relatively speaking بيكون كثير صعب كثير. يعني one of the projects اللي أنا اشتغلت عليها في البي اتش دي إنه كنا نشتغل على a natural compound، كنا نعمل semi synthesis simple changes تقريبا بجوز من 30 reaction اشتغلتهم 2-3 زبطوا، الباقيات بصير decomposition للكمباوند، بصير في interaction أو بصير في interference من ال many functionalities اللي بتكون موجودة عندي على الكمباوند مع ال reagents اللي بتنضاف. ف that is why you would see a number members of these let's say big groups قليلة جدا في الليترشر، لأنه كثير صعب إنه ينشغل عليهم، كثير صعب إنك تعملي synthesis و derivatization كيف ما أنت بدك. Alright. Any question up to this point? طيب with that yes. عفوا. لازم. يعني I don't know صراحة. هلا مش انهم they wanted to have that، بس لانه اصلا الاريثرومايسين والديريفاتيفز من الاريثرومايسين already هاي النيتروجين موجوده. هلا ما بعرف اذا انشالت كيف ممكن تاثر على الاكتيفيتي. You can judge that in theory اذا اندرس for instance exactly الinteraction of those drugs inside a ribosome. هل إنه هاي النيتروجين بتعمل crucial interaction ولا لا؟ If this is the case, then that nitrogen or that tertiary nitrogen would be very important for the activity. إيش عفواً؟ I cannot let's say يعني it seems not، بس يعني حتى كمان مثلا لما تطلعي على التليثرومايسين والسيليثرومايسين they've replaced it with another heteroatom. Yeah, yes, it's part اللي على position 12 yes، واللي على position 11 they've substituted it with a nitrogen. هلا still those can form hydrogen bond interaction weak يعني مقارنة ب يعني very weak I would consider. يعني ممكن تحكي انه مثلا يعني they may form a strong hydrogen bond interaction، بس مثلا بالتليثرومايسين والسيليثرومايسين صح انا شلتهم بس انا ضفت other functionalities that may form other interactions وتعوض عن هاي الهيدروجين bond interactions. لكن بالاريثرومايسين itself لو انا شلت هاي الهيدروكسيلز وبس ما حطيتش شيء بدالهم ممكن انه الاكتيفيتي تقل بشكل كبير، وارد. ف because of the lack of خلينا نحكي او انه صعوبه الاكسس على so many derivatives، عشان هيك structure activity relationship لهاي الكومباوندز مش let's say well established. في few let's say functionality بنعرف انها important، لكن this does not mean ان other functionalities ممكن ما تكون very important. ممكن يعني حتى اعرف 100% بدي اشيلهم one by one عشان اعرف. Any question? Good. طيب هلا المحاضرة الجاي زي ما حكيت راح تكون تكملة للبروتين سينثيسيس انهبيتورز، وراح نحكي عن التتراسايكلينز. التتراسايكلينز راح يكون في هيك مجال ألطف إنه نحكي عن structure activity relationship because those were well studied. وبهيك بنكون كملنا البروتين سينثيسيس انهبيتورز. الأسبوع الجاي راح تكون آخر محاضرة الانتي فانجلز والانتي فايرالز. الانتي فانجلز راح انزل ريكوردينجز جاهز لإلها، بس برضه بالمحاضرة راح I will let's say go through everything scan everything، بس يعني I hope انكم تكونوا at least يعني شايفين الريكوردينج in case لانه يعني مش راح ابلش فيها from scratch، يعني I will go over the very important things behind subject. تمام؟ اللي موجودين اونلاين في اي حدا عنده اي سؤال؟
يعطيك العافية دكتورة.
يعطيكم الف عافية.